Our lecture today is post-translational modification, and we're going to study this phenomenon looking at p53, which is called the guardian of the human genome. Our objectives are as follows. Number one, we're going to look at the role of p53 in the cell cycle. We're going to look at p53 regulation and some of the functions that are attributed to p53, and then we're going to look at 5 specific post-translational modifications, which is a follow-up of your post-translational modification lectures. We begin by looking at the role for p53. The reason why this protein is called the guardian of the genome is simply this, that in order for a cell to be replicated, everything must be right to go from one stage to the next, from G1 to S, G2 to M, before it begins to divide in mitosis, that everything else should be done correctly. Now, if there's something wrong with the DNA, then p53 increases in activity and it begins to arrest cell division, and it begins to stimulate repair of DNA, and if it cannot repair the DNA, then it destines the cell to apoptosis or programmed cell death. Now, in order for the cell cycle to occur, it needs the activation or activity of a number of kinases that are cyclin dependent. And they are CDK4/6, CDK2, and CDK1, and these are all cyclin-dependent kinases that stimulate the cell cycle to go from one stage to the next. p53 regulation is very, very simple, and that is that what we understand about this is that p53 is regulated at a very low level by binding to an MDM2, which is a ligase enzyme, one of the ligase enzyme that is responsible for ubiquitin. And so it turns out that p53 tetramer will bind to the upstream region of MDM2 gene and actually increases transcription of that gene. Now, transcription will lead to translation of MDM2, which will complex with p53, and it will cause the ubiquitination of p53 and destine that p53 for proteasome degradation. And so the level of p53 is controlled by the activity of MDM2. Now, these changes, when there is an insult that is inflicted upon the cell, the p53 activation will go up. Now, what happened is, and we're going to look at it, that one of the events that occurs is that p53 gets phosphorylated, MDM2 gets phosphorylated. So now the complex between the two is not formed anymore. p53 tetramer then turn on a number of genes, including the gene for microRNA 605. Now, microRNA 605 is used to degrade through degradation, degrade the messenger RNA for MDM2, thus lowering the level of MDM2 and not making it available for complex formation with p53. p53 level then is elevated. And so this is how p53 is regulated. Now, specifically, you can look at it this way. So when MDM2 or MDMX complex with p53, p53 then causes MDM2 or MDMX then causes ubiquitination of P53. P53 will then be destined for degradation in the proteasome unit. Now, in the presence of an insult, let's say radiation, when the DNA is damaged, then MDM2 is phosphorylated, P53 is phosphorylated, and also acetylated, and the complex is not formed anymore, which means that P53 is free to do its job in either arresting the cell from going through the cell cycle and causing senescence or stoppage of cell division or cell proliferation, or if DNA repair cannot be done, then it destines that cell to programmed cell death right here. So, in the absence of that complex, P53 is stabilized and it can induce all of the following. So, how is the cell cycle arrested then? Well, it turns out that you remember the cyclin-dependent kinases are required for the cell to go from one stage or one phase to the next. Now, what happened is in the presence of high concentration of P53, P53 begins to act as a transcription factor to turn on the expression of a number of genes, and some of these genes, P16, P21, P5, and P27, these are all cyclin inhibitors, cyclin inhibitor and cyclin dependent kinase inhibitors. So P53 will turn on the expression of these inhibitors, which will inhibit the cyclin and cyclin-dependent kinases, and it will arrest the cell from going from one phase or one stage to the next. Also, one of the level of control is that through the retinoblastoma protein or retinoblastoma gene project, and that is that in the presence of cyclins, cyclins will actually phosphorylate retinoblastoma. Retinoblastoma then will liberate. It actually exists in a complex with elongation factor 2, and so when it is phosphorylated, when retinoblastoma product is phosphorylated by cyclin and cyclin-dependent kinases, it will release the elongation factor. Elongation factor then is free to act or to play a significant role in DNA synthesis in the S phase. But in the absence of cyclin activities and cyclin-dependent kinase activities, then retinoblastoma is not phosphorylated, and retinoblastoma will be in a complex with the elongation factor. That elongation factor is not free to be used in the S phase, and thus arresting cell division. And so this is a very clear diagram or cartoon of the activity of P53. So with DNA damage, P53 is increased because now it's phosphorylated and it is liberated from the MDM2 protein. It's not in a complex with it anymore, so the level goes up, and then now it acts as a transcription factor for a number of inhibitors, including P21, which is an inhibitor of cyclin-dependent kinases. When it inhibits the activity of cyclin-dependent kinase activity, then it cannot, the cyclin-dependent kinases cannot do their job by phosphorylating retinoblastoma protein right here, and other jobs that are required in different phases of the cell cycle. And so in the absence of these, because they're inhibited by inhibitors that are turned on by P53, then the cell's cycle is arrested. Now, so P53 then plays a role in stimulating senescence and apoptosis, and this is just another cartoon to say exactly what we've been saying, and that is that with low concentration of P53, it is in a complex with MDM2, and then so the level of P53 is going to be low because MDM2 is going to cause ubiquitination of P53 and destine it for, sends it off for degradation by the proteasome. Now, in the presence of DNA damage, then phosphorylation of P53 occurs. This complex doesn't exist, and P53 then can stimulate the inhibitors of cyclin-dependent kinases, which will arrest cell cycle, and then it can also stimulate pro-apoptotic protein, such as BAX, to cause apoptosis. So, this is how P53 acts to regulate the cell cycle. Now, I would like for us to look at P53 post-translational modification. And the reason why this is very important is because we have studied post-translational modification of proteins, and this is an example of what happens to protein where they're post-translationally modified. And these are some examples. You can see that this is the P53 protein that has a number of areas here, transactivation domains, right here, from amino acid 1 to 100. There's a DNA binding domain from 101 to 300, and then there's a regulatory domain. This is actually a tetramerization domain right here in blue, and then the regulatory C-terminal domain that is in red. Now, you can see that this entire protein is post-translationally modified in these different areas by acetylation, or we can just go here by phosphorylation, acetylation, ubiquitination, neddylation, sumoylation, and methylation. And so we're going to look at each one of these six very briefly because it's going to take too much time and we only have 1 hour of lecture. And so we're going to look at each one of them briefly, how they occur and what role they play in controlling the function of P53. And so specifically, P53 is modified by phosphorylation in a number of residues that consist of serine and threonine, both in the transactivating region, some in the DNA binding region, and even in the transactivating and regulatory, tetramerization and regulatory region right here. Ubiquitination, neddylation, and sumoylation actually occur in different areas, mostly in the C-terminal domain of the of P53, in the tetramerization domain and in the regulatory domain. Sumoylation is the one that is very simple, and it occurs only on one lysine 386 of the regulatory domain. And then acetylation occurs on lysine right here, a number of lysines in the DNA binding domain, and also in the tetramerization and regulatory domain of the C-terminus. And then methylation is another post-translational modification that also is specific for the tetramerization and the regulatory domain. So you see this number of post-translational modification, 6 of them, that actually occur on the P53 protein. And we're going to use these as an example, and we're going to look at the effect that they have on the function of this very important protein. And so let's look at each one of these processes briefly. Acetylation is the addition of acetate group to lysine residues. And you can see, we've already talked about this, that acetylation of lysine residues on histones. And this is the nucleosome. You remember nucleosome consists of histones where DNA wraps around it. And this is the compact structure of chromatin. And so when chromatin is acetylated, it's loosened up, because this is the area that you want to do transcription or even DNA replication. So acetylation loosen up the nucleosome structure and expose the DNA, whereas deacetylation makes it compact. So it turns off the gene transcription, and acetylation turns on gene transcription. Phosphorylation is the addition of phosphate group, this phosphate group, to serine, threonine, or tyrosine residues, which can alter the function of a protein tremendously. Methylation can occur on histone protein as well as DNA itself. Methylation is just the opposite of acetylation, and that is when DNA or the histone is methylated, it leads to inactive transcription or shutting off transcription right here, because the chromatin is more compact, whereas the demethylation will open it up and make it more active, because it's less compact. The more compact one is called heterochromatin, and the less compact is called euchromatin. Ubiquitination is the addition of a short peptide molecule that consists of 76 amino acids to proteins, and when it tags these proteins on the epsilon amino group of lysine, it destines the substrate to be degraded by the proteasome unit. It is very similar to Neddylation, again, addition of peptide that is very homologous to ubiquitin, and Sumoylation is exactly the same. They're very similar, addition of peptide to substrate, but the difference between the three is that ubiquitination destines a protein to be degraded by the proteasome unit, while Neddylation and Sumoylation simply alter the activity of the substrate. All right, so this is P53. Let's talk about P53, post-translational modification. So when you do post-translational modification of P53, then you can actually alter its function so that it will either cause cell arrest, apoptosis, DNA repair, senescence, and autophagy, or it can also inhibit metabolism, angiogenesis, migration, and metastasis. So it is a surveillance protein because it plays so many functions in the cell by maintaining the integrity of the cell or the cellular environment. Let's begin by looking at acetylation. Acetylation is done by 5 different acetyl transferases, and they're called TIP60, PCAF, MOS, MOF, and P300 and CBP. These are the histone acetyl transferases that play the role of transferring acetyl group to lysine residues on P53. And there's a number of domains, again, transactivating domain, DNA binding domain that has 2 lysine residues that are acetylated, and then you have the tetramerization domain, and then you have the C-terminal regulatory domain that also has a number of residues that are acetylated. Okay. Now, what is the effect of P53 acetylation? Well, acetylation does a number of things, 3 things. Number 1, it promotes P53 stabilization by excluding ubiquitination on the same side. In other words, if you have acetyl group that is acetylating lysine, then it prevents the addition of ubiquitin to that residue, and so it will prevent degradation of p53 if that occurs. The second thing that happens by acetylation is that it inhibits the formation of HDM2 or HDMX and p53 complex. In other words, when you acetylate p53, p53 doesn't exist in a complex with the HDM2 enzyme anymore. And so its activity is not repressed. And the third one is that it recruits cofactors for the promoter-specific activation of p53 for increased transcriptional activity. So, in a nutshell, acetylation of p53 increases its activity as a transcription factor. And so you can see here there are two cases that we're going to show you. So acetylation of p53 then, you see acetylation of the C-terminal domain causes a conformational change here. So acetylating it, conformational change, and so it opens up and it prevents occlusion of the DNA binding domain. In other words, now that you've opened it up because you've acetylated, now p53 can bind to DNA as a transcription factor. And the second thing is that when you acetylate, especially on lysine 120, here's what happens. You turn on the P21 inhibitor right here, expressed on P21. It causes cell arrest. It also stimulates cell death. It also stimulates senescence, again through the P21 inhibitor as well. And of course, it stimulates cell death. So it arrests cell division. It causes quiescence of the cell, that it doesn't enter into the cell division or cell cycle anymore, and it also destines that cell to die. And so that's these are the effects of acetylation of p53. And you can look at this table, but the consequence of acetylation is that without acetylation, cells survive and cell continues to proliferate. But in the presence of acetylation, you arrest the cell cycle, you put the cell out of the G1 into the G0 state, and then you destine that cell to die by programmed cell death. So acetylation changes the function of p53 tremendously. Phosphorylation. Phosphorylation, there are a number of residues that are phosphorylated on p53, and they are located in all of the region, and so phosphorylation occurs on serine and threonine residues. Okay, we'll talk about just a number of these, four of these serines, and so serine 15 and 20. And when they're phosphorylated, they reduce p53 affinity for HDM2. In other words, phosphorylation on serine 15 and serine 20 will reduce the affinity of p53 for HDM2, which means that it's going to free up p53 to serve its role of increasing transcription of substrate genes. If you were to mutate serine to alanine on both 15 and 20 residue from serine to alanine, these mutations will lead to defective apoptosis and senescence, and they will lead to development of latent tumors. So that's by changing the ability of these residues to be phosphorylated, you actually increase the likelihood of developing latent tumors. Phosphorylation of another residue, residue number 46, induces pro-apoptotic genes. So this is very important. Again, phosphorylation of serine 392, which is in the regulatory region, in response to ultraviolet light, stabilizes p53 tetramer and induces gene transcription. So in response to ultraviolet light or DNA damage, it's going to stabilize the p53 tetramer so that it can serve its role as a transcription factor to elevate the transcription of a number of genes. So if you were to mutate that serine to alanine at residue 392, then that mutation predisposes that cell to ultraviolet-induced skin cancer, or in humans or in animal models. If you were to do that mutation in p53, it predisposes that host or that animal to ultraviolet-induced skin cancer. Methylation also serves a critical role in post-translational modification of p53. Methylation occurs on lysine, which is K, and arginine, which is R. Again, it occurs in the tetramerization domain and the C-terminal regulatory domain. So it can either be, methylation can either cause p53 to be activated or to be repressed. For instance, methylation on residue 372 promotes transactivation of target genes. So methylation of lysine 372 turns on the activity of p53. Methylation of 382 by a single methyl group or 370 by a single methyl group will repress the activity of p53. Okay, it will inhibit its ability to serve as a transcription factor. Dimethylation, however, of lysine 373 will repress the activity, but dimethylation of the residue that we've looked at, monomethylation of lysine 370 will depress the activity of p53, while dimethylation of the same residue, lysine 370, will stimulate transcriptional activity in response to DNA damage. Again, this is a very unusual one because post-translational modification by methylation either turns on p53 activity or turns it off. Now let's look at ubiquitination. You remember we say that polyubiquitination will destine a protein for degradation, but monoubiquitination is usually used as a control of activity, protein level. So polyubiquitination by high levels of HTM2 and E3 ligase targets p53 for degradation, while monoubiquitination by low levels facilitates p53 cytoplasmic translocation, and it serves as a steady state control of p53. Again, high ubiquitination will degrade the p53, while monoubiquitination will serve to control its steady state level. So suppression of p53 activity by HTM2 serves two things. It targets it for degradation by the proteasome unit, and it inhibits transcriptional activity by directly binding to and repressing p53. Okay, so again, polyubiquitination is used to control the activity of p53, either poly or monoubiquitination. And so, as we say, HTM2 controls the level of p53 by either polyubiquitinating it and destining and sending it off to be degraded by proteasome, or by binding to it and preventing it from binding to its side on the upstream region of a number of target proteins. Sumoylation and Neddylation, we're going to combine these two. Again, remember that they're very similar to ubiquitination in that this is the addition of peptides that are highly homologous with the ubiquitin to lysine residues. And there's only one site for sumoylation, and that is lysine number 386. While there are 5 sites for neddylation, lysine 320, 21, 370, 72, and 73. Now, look at this. Neddylation inhibits p53 transcriptional activity. So it serves only one purpose, and that is neddylation of p53 at these residues will inhibit the transcriptional activity of p53. However, the sumoylation of the single lysine residue 386 will actually increase the activity of p53. In fact, the increase is so tremendous that it will actually lead to premature cell senescence. So the activity is not just increased to a proper level, it increased so much so that it actually leads to abnormal or premature cell